My name is Ian Stocks. I'm a taxonomic entomologist with the Florida Department of Agriculture and Consumer Services, Division of Plant Industry in Gainesville, Florida. The groups that I'm responsible for are the scales, mealybugs and their relatives, and the Allarodidae, which are the whiteflies. So we got the key, got the illustrations, the plates for the soft scales, and I think the first one we'll start with is Milvascuculus mangiferi. Okay. And what we'll do is we'll just take a general tour of the morphology at a couple of magnifications of this particular species, and we'll pick out some characters that are generally applicable to the other species we'll look at. So there may be some characters that we talk about on this slide that are not necessarily related to things that might appear elsewhere on the key, but we'll hit them anyway. So go ahead and get the slide set up so you've got it on the 10x. Okay, so you've got roughly the head area in view. So the front margin all the way down to where the antennae, the first pair of legs, and the mouth parts originate. Okay. All right, so we all set up with that at around 10x. So a lot of this morphology is going to look in gross scale fairly similar to that of the mealybugs. The antennae, the mouth parts are quite similar, of course. The legs, the number of segments in a leg as you move down the body. You pick up the spiracles, the first pair of spiracles to the just to the side of the first pair of legs. Okay, posterior pair of spiracles there in this slide, just halfway between the second and third pair of legs. Depending on how the specimen is mounted, you may see tubes sort of coming off from around this area all throughout the body. Very often those things come out of the specimen during processing, but at least for some species they do remain. Those are the tracheae that go throughout the body. They get sort of branch out gradually as they get further and further away. The other thing about the spiracles, let me go back to the anterior one. It's a bit more clear in this specimen here. These are on the ventral surface, and of course these animals are pretty much stuck down on their venter on the substrate. But they have to have some port, some access for breathing, getting gases to them. So what happens is that there's a channel essentially that's formed just by the shape of the cuticle here called the spiracular furrow, and it joins up here with the actual spiracle where the gases then can diffuse. These are reasonably consistently present in all of these groups, and one way to identify them is first by simply finding the location of the spiracle, and then if you see that's depending on the magnification, you can actually perceive these little dots at this magnification at least that progress up the channel. Those are a type of pore, a type of multilocular pore that secrete wax that helps keep that spiracular furrow coated and helps it stay clear from water building up. And then typically, though not in all cases, the spiracular furrow, when it joins the margin, will have marginal setae that are distinctly differentiated from any other setae that are around the margin, and we'll come back to those in a minute. Okay. Go back to the middle, and we'll just continue moving down the body here. Okay, so between the legs here on the venter, of course these are insects, so they're segmented, and you can still see parts of the segmentation pattern here. For instance, these pink lines are sort of enhanced sclerotization that mark the boundary between two segments. Okay, we'll continue down. Now we're going to get to the key feature that you want to look for when faced with a slide of possibly a soft scale. This is a character that is diagnostic for the soft scales. These are the anal plates. Okay, each one is an operculum. Together they form the opercula. So they're a paired structure. They're by and large, roughly some version of a triangle in outline. And this is a fairly extreme version of how distorted that triangle can be. And the exact proportions of the size of this triangle in this case are characteristic of this genus. Okay? Now, associated with this anal plate is the structure here, which is the anal tube. Okay? And that's the structure that ejects the honeydew droplet. Because the anal plates, they form, they're paired, they form a kind of a slit-like structure that continues down the body until it gets to the margin here. Okay? That's the anal cleft, and the length of that can be important as well for diagnosis. So we'll continue, and we're going to go pick up a leg. Let's go pick up a leg now. Any leg will do. Okay. This is another structure that can be quite variable at the genus level and at the species level. For instance, there are genera that have some species with very well developed, fully articulated legs, and then there are other species in that genus where the legs are reduced almost to nothingness. This is a case with a fully developed leg. As we did for the mealybugs, you can go to the segments. This is the coxa here, the first segment that articulates with the body surface. The next segment down is the trochanter. Then we have the femur. The tibia here. And then angling down is the last segment with the tarsus. And that ends in the claw. And like they have in the mealybugs, there are these paired structures that kind of look a little bit like spatulas. They're the digitules. This is the tarsal digital pair. The tarsal digital pair is the long pair here, and the claw digital pair is the one that is actually more spatulate. Now, one of the most important characters in the leg for soft scales is the condition of this joint here, the tibia and the tarsus. You can see right here, in this specimen, there's actually a very darkened kind of knob in there. That's called the tibio-tarsal sclerosis, or articular sclerosis, okay? Now, this has several kinds of, this leg has several kinds of conditions. One is it has that well-developed, and the leg is able to bend a little bit. Okay, that's a tibio-tarsal sclerosis with full articulation. There's another case where they have that, but the leg is still more or less kind of fixed in one place. Okay, so that's tibio-tarsal sclerosis without articulation. And then in one of the other slides we're going to look at, there's no one there at all, and the leg is actually essentially one fused long unit. Okay, so we always will have to evaluate that condition. We're going to move to the margin, the body margin. There's a tremendous diversity of form in the seta that go along the body margin. In the case like this, for instance, they're fairly short and fringed. Some cases they are quite long, and they can either be acutely pointed, bluntly pointed, fringed at the tip and long, just tremendously variable and usually consistent at the species level. Now, if you go back up the body margin to pick up see if I can find a spherical here. The marginal setae throughout most of the margin of the specimen are pretty consistent to their general morphology, and I mentioned this earlier, but when you get to the spiracular furrow, where that joins the body wall, frequently you'll find them that will have a different morphology. And a very common case is for there to be a set of 3, a pair that are fairly similar, flanking a single seta that is distinct in its morphology. Okay, so it'll be usually longer or have some kind of other shape development to it, but they typically will stand out. There are a few genera where they are all the spiracular setae are undifferentiated from the marginal setae. Okay, and I'm now moving on to the go down in magnification here, back onto the surface of the body. We're going to pick up some of the features on the general sort of body cuticle here. So I'm going to go up to 400x here. And one of the characters I'm looking for towards the body margin here, go back out. Going to get a moment here to find some good examples. Here we go. Here we go. The dorsum is going to be ornamented in many cases with various features. Some of them are duct-like in their function. If you go to the body margin and you progress along until you pick up on one of these they're basically a circular bump. Okay? Let me see if I can get that out a bit more here. This is a dorsal tubercle. These can vary at the species level from 0 to 10 or more. And even within a species in a population, you can find some specimens that have them, a number along the margin, and then some other specimens where they have none. They're restricted to the body margin when they are present. There is a little bit of micromorphology about their actual structure that can be important, but it can be a difficult structure to get much detail on. So we'll run across those further in the key. Now, if you do move on to the more into the center, pick out, if you can, some of the dorsal setae. In this specimen, there'll be a prominent socket and a fairly short, stout setal structure that's kind of clubbed, a little bit clubbed. It kind of gets expanded towards the tip a little bit. Okay. Can you see that in this one right here? That's characteristic for this genus, but those dorsal setae can vary to being very conical and stout. Fairly short, pin-like. Again, just a tremendous amount of diversity, but fairly consistent at the level of individual species. And there's various other ornamentation types too, like pigment color patterns. So you can see there are various clear spots here, changes in staining intensity, reflecting differences in levels of sclerotization, but those are generally not a character that we would be interested in, except for a few cases where they're so distinct that they actually are part of the diagnosis. So before we move to the key, let's just quickly put in another species and just draw some comparisons. Pick up the one for Coccus hesperidum. We'll go back to 10x. Okay, are you all set up with that slide? You can see it at 4x, but we'll go ahead and go to 10x. If you can go towards the posterior of the specimen and look for the anal plates, okay, and find them and go to 10x. Now we're on plate 7. I just wanted to show you the difference between the shapes of the anal anal plates between the first species we looked at, the Milvascuculus, and this one, which is the brown soft scale. Again, you can see that they're both generally sort of triangular in outline, but of course the margin on the brown soft scale compared to that for the previous slide is much shorter, so it's a more regular kind of triangle. There could be some curvature along a margin, and these angles could be rounded as well to some degree. But the general outline of the shape is going to be consistent at the species level. Okay. You can also see the anal tube here. There's the opening for the anal tube. All of this in here is the membrane that surrounds it, and these sclerotized regions in here are the anal tube setae, or the anal ring setae. Okay. So this structure is an aversible tube. It can be pushed out of the interior of the body under pressure. The ring at the end of it has setae on it, and the idea is that the droplet of honeydew is pushed out along that tube, gets to the end, and is kind of held in place by those anal ring setae. Okay? And then when the muscles contract down on it fairly rapidly, those setae close up, and it ejects that honeydew droplet away from the body. So it's not just a shape or a piece of morphology, it's a piece of functional morphology helping them to eliminate the honeydew. Okay? All right. And I just want to make one more comparison for a moment here. It's going to be the slide for Sycetia coffeei, and it's going to correspond to plate 3. So if you can find the anal plates again. You get those in the center at 10x. See if you can go to 20x. There's a character here that's important for diagnosing this genus, and this is a worldwide genus. We have in Florida, though, we have about 4 or 5 species, but it's also a well-traveled genus, so species occur around the world and are frequent in trade. A way of diagnosing this genus pretty rapidly is, if you look at the anal plates again, they're adorned with setae. Almost all of these will have some kind of setae on them in different positions, but this one has a very characteristic seta here called the discal seta. It's a very prominent, well-demarked socket, usually with a very stout seta attached to it. Okay, so we'll come back to that. So there are features on the anal plates that are very useful for either genus or species level diagnosis. Okay. This is also another species that has tibiotarsal sclerosis with full articulation. Okay.